Good afternoon to everybody. As a Vice President of EIPG, European Industrial Pharmacists Group, I'm very pleased to open this webinar, which has been organized with the technical assistance of Tecniche Nuove, which is an Italian publisher, partner of EIPG since a few years, especially for the issuing of our monthly newsletter, which is sent to all our European members. This webinar has also been possible for the support of two sponsors, two companies which are well known on the market, offering technological solutions which are applied in the manufacture of sterile products. In particular, we have Lama, which is a historical, successful Italian company, founded more than 70 years ago, with high competencies in the manufacture of sterilization equipment. And another company is a Commissar, originally an Italian company, since 2 years ago part of a multinational group called ATS Automation, a Canadian group which is specialized in containment but also in automation. The purpose of this webinar, as reported in this front slide, is a focus on main issues which came up in the last version of the Annex 1. Let me show you the webinar plan. Because of the organization, we have, I will spend a few words introducing this webinar. Then I will leave our speaker, I will introduce very soon, Francesco Boschi, to outline the main comments of Annex 1. Then we will have a first question time. Then I will invite the two sponsors to present some interesting, innovative technical solutions which can be applied, of course, in the sterile medicinal products manufacturing. Then we'll have a second question time so that all participants can again send their questions to the speakers based also on what they had seen from sponsor, and then some conclusions. The total time of this webinar could be around 1 hour and 10 minutes. Let me start right now spending a few words about EIPG, which is a European association representing the national professional organization of pharmacists employed in the pharmaceutical and allied industries in the member states. And EIPG was among the 16 organizations which were recognized by European Commission as stakeholders to take part in the consultation of Annex 1. So EIPG worked in a team with the other organization for an exchange of ideas, observations of the last version of Annex 1. Then it was up to each organization to prepare its own technical comments, which were requested to be submitted to European Commission. And this is the focus on this webinar. So the technical comments the EIPG prepared and submitted. But next to this, it is to be underlined that EIPG also signed a joint letter with the other organization, which was addressed to European Commission just a few days before sending the technical comments. This joint letter contains a few general considerations about Annex 1, and I have summarized in 3 lines the main focus of this letter, expressing the need of flexibilities to support the use of alternative approaches. This is a point which will be reminded of during the presentation of our speaker. Then another point highlighted in this letter was the need of a clearer interpretation of a few critical points of the Annex 1. And then the same letter emphasized how it is important to keep a good dialogue between regulators and industry to promote a better understanding of Annex 1. So just before letting Francesco Boschi to outline these technical comments, let me say that it was the real important contribution given by an Italian Association of Industrial Pharmacists, AFI, which prepared a first draft document. This draft document was circulated among all delegates of the EIPG and was the basis for preparing the final document which was submitted to European Commission, taking into consideration particularly the contributions of some experts among our members from Spain and from Belgium. So I would now leave room to our Francesco Boschi, who is our speaker today. He was coordinator of the group, but he will explain better what he did in the preparation of the technical comments. Francesco Boschi studied biology at University of Milan, but also got a postdoc degree in applied biotechnology. Spent more than 15 years in the quality assurance, quality control positions in the area of industrial microbiology and sterility assurance with the important multinational companies. He is currently part of the Pfizer Global Microbiology and Aseptic team, supporting the sterile injectable network of Pfizer Global Supply. So I would invite Francesco to start his presentation.
Thank you. Thank you, Piero. So good afternoon, everyone. Today we are going to talk about the comment collections on the draft Annex 1 version 12, which is the last Annex 1 version published. And we will highlight the most important concepts that have been commented by EIPG. So yeah, Piero kindly introduced me. Anyway, yes, I'm part of the Microbiology and Aseptic support team, which support globally the Pfizer supply network. So I have a quite wide experience in pharmaceutical microbiology and sterility assurance. And also since 2010, I am chairman of the AFI Microbiology Interest Group. And this is my contact information. So if you have any question also after this webinar, please don't hesitate to contact me. So which will be the content of our presentation? First, we are going to see the EU GMP Annex 1 and its revision process. Then we will have an overview of the content of the draft Annex 1 version 12, which have been published this year. And for each of these points, we will review the most significant comments which have been collected within EIPG. And finally, we will have a look about how within AFI and EIPG, we collected the comments even in this difficult pandemic situation. So, Annex 1, we all know that Annex 1 is an appendix of the Utralex Volume 4, the good manufacturing practices in the European Union. And particularly, Annex 1 is focused on the manufacture of sterile medicinal products, both terminally sterilized and aseptically filled. Annex 1 was first published in 1971, so it's almost 50 years old. So it's quite an old document. And of course, throughout this time frame, it has undergone through a number of targeted updates. And the last one was published on February 14th, 2008. However, this is the first time since 1971 that the document has undergone a full review, because this is not just a simple review. It's more that the document has been completely rewritten and expanding in many, many different concepts. So, according to the EMA website, This revision is intended to add clarity, to introduce the principle of the quality risk management, and this will be a concept that we will encounter many times during this presentation, to allow for the inclusion of new technologies and innovative processes, and also to change the structure of the document itself to allow a more logical flow. So the key changes, again, according to EMA, are the introduction of new sections. We have brand new sections like the utilities section, which is not present at all in the current annex in the 2008 version. The introduction of the principle of the quality risk management. The document, again, was restructured to give a more logical flow, and it's much more detailed. So it's about 50 pages, so more than double than the current annex. So many concepts has been treated in more detail in respect of the current version. And that's why this document is so important. One of the key concepts of the new draft Annex 1 is the contamination control strategy, which is an overall strategy which embraces different controls to ensure the aseptic process, especially the aseptic process, is maintained under control. So the plan for the CCS is first to define all the critical control points. So throughout the process to identify where a microbial contamination risk can be present, to assess the effectiveness of the control that are implemented to manage this risk, which from every point of view, from a design point of view, procedural point of view, technical and organizational point of view, and finally to monitor the measures employed to manage the risk. So define the control point, assess the effectiveness of the control, and monitor. The CCS, of course, is not a static document, should be actively updated because our manufacturing processes are never static. They are always evolving, always changing, and should drive to continuous improvement. And this is another important concept that has been introduced in this draft, to have a continuous improvement of the manufacturing and control methods. So which is the history of this current revision process, which have been running for quite a long time now. The first draft was released by EMA at the end of 2017, in December 20th, and then a first targeted consultation was performed. And this involved about 140 companies and other organizations like PDA, EIPG, and so on to comment. This revision process was quite a success, maybe a little bit unexpected by EMA, because they had to process more than 6,200 lines of comments, so a huge number of comments. And about this first consultation run, both AFI and EIPG contributed, and particularly AFI issued 3 general comments on the annex and about 40 comments on specific points. And we were very pleased to see in the new draft then several of the comments issued by AFI, of course, probably not only by AFI, but anyway, something that we consider as AFI, are now reflected in the new draft. So hopefully it will be the same also this time. Because now a second draft has been published on February 2020, so this year, and the second targeted consultation has been already completed. So it was from February, it was supposed to last until May, but then it was postponed due to the COVID-19 situation to July 20. Only selected associations, including EIPG, have been involved in the consultation. But also, in order to maintain a global alignment, according to EMA, in parallel, the same document will be subjected to a second targeted consultation by WHO and by PICS. So from this, we can see that this annex will be probably the most important regulatory reference throughout all the world in the next few years. So not only in Europe, but probably it will influence also GMP regulators and inspectors throughout the whole world. So maybe it's not very easy to read, but these are the part of the Annex 1 which have been specifically open for consultation, and we are going to see all of this very quickly. But also this was not limited, but also because also EMA allowed the possibility of issuing comments on every other session of the annex, with the only limitation in the last line we can see, please avoid resubmitting comments which you have already submitted at the first consultation, of course. So these are the chapters which have been open, specifically open for consultation. You can see in the slide, and these are the most important chapters in the annex. And I'm not going to spend much time on this slide because we are going to see one by one. So the first chapter is chapter 4, premises. And this section describes the different facilities where the aseptic process can take place. And the section commenting about the definition and handling of barrier system, which are going to be the new standard now for the aseptic manufacturing, and the qualification and requalification of clean room. And by EIPG, we issued a comment, for example, at point 4.3, and specifically related to the part where the annex states that any alternative approaches to the use of wraps and isolator should be justified. And our comment was the sentence starting with any alternative approach may be misinterpreted. So we suggest to clarify how alternative approaches can be supported, especially in existing facility, because many companies have in place conventional clean rooms right now. So it should be clarified how this can be supported, at least for the short term. So our suggested text was any alternative approaches to the use of wraps and isolator, especially in relation to existing facilities, should be supported by a documented risk assessment considering historical data and established control. So we tried also to put some elements that can help the different companies to write this risk assessment, which of course is needed now to support any other facility different than wraps and isolator. Because now it's very clear the annex is putting the standards towards the isolation between the operator and the process, the separation more than the isolation. Another point that we commented is point 4.23, and this is about the sterilization or decontamination of wraps gloves. And particularly the annex say that wraps gloves using a grade A zone should be sterilized before installation, and sterilized or effectively decontaminated by a validated method which achieve the same objective prior to each manufacturing campaign. Our comment that the requirement of performing decontamination of wraps gloves by a validated method which achieve the same objective of sterilization is ambiguous. So is a 6-log reduction required? We think this should be clarified, because 6-log reduction can only be achieved by automated processes like VHP, which may not be feasible in open wraps. So how can we see this requirement in open wraps, for example? And we think as a team from the discussion and a validated sanitization, ensuring a 3-log reduction of vegetative cells and 2-log reduction of spore may be typically adequate to guarantee that the gloves are free of contamination prior to the manufacturing campaign, because typically the initial contamination of these gloves is quite low. So we suggest to reward as wraps gloves using grade A zone should be sterilized before installation and sterilized or effectively decontaminated by a validated method prior to each manufacturing campaign. So we suggest to remove the reference to the same object as sterilization. 4.29 is another point that we commented, especially about this suggestion to consider the use for clean room classification of larger particles in respect of a 0.5, for example, 1 micron particles. So as a comment, we stated that we appreciate the removal of the 5 microns for grade A and B at rest classification, as this allows harmonization with the ISO 14644/1 table 1. However, this introduces a discrepancy with the monitoring recommendation, where the 5 micron particles have been retained. Also, by this wording, it is not clear if this is a requirement, a recommendation, or an expectation. And what we think is the need of considering larger particles should be assessed within the contamination control strategy, considering the specific process and the specific facility. And if this will be the case, so if considering larger particle is needed, we recommend to refer to the ISO 14644 table, where we have all the limit and all the sites which needs to be considered. So the rewarding is for grade A zone and grade B at rest, classification should include as a minimum the 0.5. If the need of considering larger particle is identified within the contamination control strategy, refer to the ISO 14644/1 table 1 for particle size and limit. So this is the rewarding that we suggested. Let's see if it will be captured or not. Also, the table 2 was commented by us. The table 2 is the one with the limit for the viable environmental monitoring in the different classification. And we commented specifically this note 2, where they say that the limits are indicated in CFU, but if different or new technologies are used that present results in a manner different from CFU, the manufacturer should scientifically justify the limits applied and, where possible, correlate them to CFU. We appreciate very much this reference because sometimes the rapid methods have a sensitivity greater than the traditional method, so it makes no scientifically much sense to refer limit in CFU. But what we didn't still appreciate is this reference of the correlation to the CFU, because this may be not possible for some method, for some other maybe not scientifically supportable. So we recommend to apply the science most of all, rather than referring still to the traditional method. So what we say, what we should recommend, that if different or new technologies are used that present results in a manner different from CFU, the manufacturer should scientifically justify the limits applied, so without a correlation with the CFU. Let's move now to chapter 6. As I was mentioning before, this is a brand new chapter, and the comments were about the handling of water system and the handling of gas filters. One comment was about that after disinfection and regeneration of the water system, monitoring, of course, should take place, and the results, according to the annex, should be approved before the water system is returned to use. But for the traditional microbiological method, this would imply to wait for more than 5 days. So we think as a team that this requirement may be excessive, especially for qualified and well-known system. So what we recommend is to reward like data should be available and evaluated by the quality assurance unit before the water system is returned in use. Results should be approved before release of impacted batch. We think this approach may make sense and also can guarantee the final quality of the release batch. Personnel was open for consultation as well, and specifically personnel qualification and gowning. And we commented especially this point 7.6, and this is about the access of unqualified persons to the clean room. And specifically this part where the annex stated access by this person, by the unqualified person, should be assessed and recorded in accordance to the pharmaceutical quality system. An authorized person from the manufacturer should supervise the unqualified person during their activity. And what we say that we think that the access of unqualified person should never be allowed in presence of exhaust product and critical components and surfaces. And the supervision, which is suggested by the annex, we don't see as an effective mitigation for the contamination risk, for the risk that this unqualified person could spread contamination into the clean room. So our suggestion is the access by these persons, again, the unqualified persons, should never occur in presence of exposed product or critical components and surfaces, and should be assessed and recorded in accordance with the pharmaceutical quality system. Also, we have this is a very specific comment about the sterile face mask, which is now required to be worn in the clean room, and generally it makes perfect sense, of course. But for high potent products, Sometimes manufacturing respiratory protective masks are needed for the safety of the operators, and many of them cannot be sterilized. So we recommend to say that a sterile face mask should be worn, of course, but when sterilization is not possible, for example, respiratory protective mask, a validated decontamination process should be applied on these items prior to gowning. Chapter 8 is the longest section, maybe the most important section of the annex, because it goes through each different technology which is now applied in the manufacturing of sterile product. And several, as you can see by the slide, several sections have been open for consultation. And our comments were referred, for example, to point 8.41, which is a little bit ambiguous because it says that suitable BIs placed at the appropriate location should be considered as an additional method to support the validation of the sterilization process. So for us, it's not clear whether the use of BI in routine sterilization cycle is recommended. But if this is the case, we think that a validated process should not be confirmed, should not need to be confirmed every time with BIs, which is the meaning of validation. And also the use of BIs, which are living cultures, in routine manufacturing, we see this as a risk. So we suggest to specify that suitable BIs placed at appropriate location must be used for the validation of the sterilization process. But we don't see the need of a continuous challenge, and we see also a contamination risk on this practice. We have a comment, of course, on the PAPSIT section, and in this version of the annex, EMA open to some more flexibility about the consideration of the real need and the risk associated to the PAPSIT. And the annex say that it may not always be possible after sterilization to run PUPSIT due to process constraint. For example, the filtration of small volumes, very small volume of solution. So we appreciate this more open statement, but we suggest to remove the example or add more examples, because we think that the small volume is not the only situation where the PAPSIT may introduce more risk than the benefits, because sometimes it can be related to a contamination risk, to a breach in the sterile boundaries. So in our opinion, this needs to be evaluated. So our proposal is it is recognized that pre-use post-sterilization integrity testing may not always be possible after sterilization due to process constraints. So removing the access, the example. And in this case, everything, of course, will need to be very strongly supported by a risk assessment. Chapter 9, this is a chapter about process monitoring. And it's very interesting that the annex put in the same chapter viable monitoring, non-viable monitoring, and media fields, aseptic process simulation. And also they say that the reliability of each of the elements of the monitoring system when taken in isolation is limited. So it's very important to consider the all elements in a comprehensive process monitoring strategy. And the section commented, open for comments, and that we commented as AFI and EIPG, were personnel monitoring and aseptic process simulation. For example, this is a comment for related to the media field, and where they say that these units, media field units, should be inspected under similar conditions to those for visual inspection that facilitate the identification of any microbial contamination. We suggest to remove the statement about the conditions similar to the product, the actual product visual inspection, because some requirements may be different in inspecting a lyophilized vials while a vial containing culture media, for example. So we think it's enough to say that the filled unit should be inspected by staff which have been trained and qualified, and under conditions that facilitate the identification of any microbial contamination, which may be the same as finished product or maybe not, depending by the specific process and product. Also, this is a comment about the number of successful media field to revalidate the line before a media field failure. And this point, which is applicable also for one single contaminant, they say that a sufficient number of successful consecutive repeat media feeds, normally a minimum of 3, should be conducted in order to demonstrate that the process has been returned in a state of control. We think that in this case, the annex should refer to the wording of the PICS guideline PI 007/6/6, which in our opinion is very pertinent because it relates the number of the repetition run needed to the investigation outcome. So the suggested text is that sufficient number of successful consecutive repeat media feeds should be conducted in order to demonstrate that the process has been returned to a state of control. Depending on the result of the follow-up investigation, this may require the inclusion of 1 to 3 satisfactory process simulation tests. And this last statement is exactly the same wording of the PICS guideline. And we have suggested to implement this wording in the annex. Last section is the quality control section. And the section open for comments have been the quality control and the sterility test. And particularly, we commented the sterility test samples collection, point 10.6, and the annex required to collect samples for sterility test after any significant intervention. For example, intervention where the integrity of the barrier is breached, open door, or an intervention where the operator is operating within the critical zones. And in relation to this test, to the last example, based on the other chapter in the draft, this can be interpreted as any intervention in grade A, especially for conventional line. So this will result in a significant increase in sterility testing. And what we think is that it's needed that each intervention should be evaluated by for its criticality, and the need of collecting samples should be managed, should be assessed based on a rationale. So our suggestion is, for products which have been filled aseptically, samples should include containers filled at the beginning, middle, and the batch, and after any significant intervention as determined within the contamination control strategy. Also in point 10.6.3, we have that for products that are lyophilized, samples taken from different lyophilization load. We think this different is a little bit ambiguous, so we recommend to specify that samples should be taken from each lyophilization load. Also to harmonize the requirement with the terminally sterilized product, where samples should be collected by each autoclave load. And finally, we have few comments on the other chapters. As I was mentioning before, we had the possibility of commenting also other parts of the annex. So we issued this comment on point 5.5, and specifically on the requirement that direct and indirect contact parts should be sterilized. And indirect contact parts are like the stopper balls, stopper track, so surfaces in contact with primary components. In existing facility, it may not be possible to remove and sterilize indirect contact parts. So for example, we have isolators, which generally are very good, well tooled to protect the product, but in some cases in existing isolator, for example, stopper ball cannot be dismantled, so cannot be sterilized. So the use of methods, validated methods such as VHP, we think should be allowed, provided that they are supported by a contamination control strategy and by effective mitigation. So what we suggest is that in existing facility, the last part of the statement, the use of a validated decontamination method which achieves the same objective of sterilization for indirect contact parts should be supported by a risk assessment and by procedure minimizing the contamination risk. So as you can see, we adopted the same wording of the annex from the point related to the RAPS class. A couple of comments on the glossary. In the annex, they are still talking about non-viable particles. We think it's not 100% correct term, because we know the particle counter can count both the viable and the non-viable, and they cannot distinguish between viable and non-viable. So we recommend to adopt the wording total particles, and this is the same comment that was issued by PDA, for example. Finally, we are close to the end. I would like to spend a few minutes in highlighting how we manage the comments collection within EIPG and AFI. So, of course, different than 2017, this consultation was fully in the middle of the pandemic emergency, especially in Italy. So it was not possible for us, of course, to organize face-to-face meeting as we did in the 2017-18 in the AFI offices in Milan. So what we did is, after the new draft publication, we left a defined time frame for each member of the Microbiology interest group in AFI to review the document and to provide comments. Then we organized a number of online meetings to discuss the different comments received and to consolidate them. AFI provided the IT support for this step. And these meetings, it's very important, were also open for members of other AFI teams, for example, from the quality assurance team, from the validation team. And this was very good because we have also this contribution, not only from microbiology, but also from other SIMEs. AFI comments were merged then with the comments from the other countries in Europe within EIPG, and especially Spain and Belgium, as Piero was mentioned before. And we can say that discussion managed in a remote way was quite effective and also allowed an active participation of members located in the whole country. Because typically for people working in the center south of Italy, it was difficult to join the face-to-face meeting in Milan. So the remote was also the chance to have a broader audience and more active participation from all the members. And finally, the comments were submitted on time to EMA by EIPG. So we can think it was a pretty successful process despite the COVID emergency. And so now let's see how many and which comments will be captured and addressed by the EMA. So that's all. Thank you for your attention. And if you have any question, I will try to address.
Thank you, Francesco, for your presentation. I think you were able to give an overview, of course, a very quick overview, half an hour, for getting all the participants aware of the efforts made in identifying comments to be sent to a European Commission in order to clarify different points. Unfortunately, what it is evident is there are some points which need a clarification. We have now 10 minutes, about 10 minutes for questions, and we have already received a couple of questions. I think that Francesco can read also on the chat, on the chatter. And one question is about the point you made on the sterilization of gloves in a grade A area. And the criticism you expressed in your comment about the need of decontamination and not sterilization, so the different approach which is to be considered. Can you comment something?
Yeah. First of all, of course, any of these are not my comments, are the result of a discussion within the team, of course. And I think this specific point is, I think more clarification is needed because this decontamination, which achieved the same objective of the sterilization, seems to indicate the expectation of having a 6 log reduction, which is feasible for isolators and closed wraps, and this is pretty the standard, while for open wraps may not be possible or may not be possible in most cases, or may require the upgrade of the wraps to a closed wraps. So I think EMA needs to first clarify the expectation. And according to the discussion, again, within the team, we think that the sanitization with a sporicidal, considering that the bioburden of the gloves which have been sterilized before installation and then retained in a grade A environment may not be so high to pose a excessive challenge for a manual disinfection. Of course, manual disinfection is still a manual process, so it's less reliable than an automated process. But of course, what we think in the team is that EMA should provide a guidance for all the potential kind of facility. Of course, if you can have an isolator closure wraps with a decontaminated system, you can offer the highest level of protection for the gloves and then for the product.
Great, thank you, Francesco. Just in brackets, there are a couple of technical questions about to get a certification of attendance of this webinar. Yes, I confirm that EIPG will issue a certificate of attendance to each participant. Another minor point is about copy of the presentation. Yes, copy of the presentation will be available on the EIPG website, which is accessible. Let's have some other questions. What I understand is that the CCS, the contamination control systems, is a real new approach. Or do you think that it could be considered as a different way of considering an activity which is normally carried out in managing sterile manufacturing? I mean, and another question, would the CCS, in your opinion, would require specific new training for the personnel involved?
Yeah, for sure, CCS is not a new concept in an absolute way, because I think many companies already approach the control and aseptic process in this way. And also, I think it's a combination. It's the recommendation of putting together all the different elements that probably are, and for sure, in a validated aseptic process are already in place. So the personnel, the equipment, the sterilization, the disinfection, the monitoring. So the CCS is to put all together, starting from the risk, starting from the identification of the potential contamination risk and where and when in the process are located, and then in adopting the measures from every point of view to mitigate this risk. So it's a recommendation, it's a suggestion to think holistically about your process and to manage this in a structured way. So in an overall document, which can be the basis then for the specific validation document. So nothing really new, but as a concept, at least for the annex, I think it's one of the most important new addition, new concepts.
Yes, and do you think that this correct, new, well-structured approach can be applied to existing facilities? I mean, if you have some, let me say, old facilities in place, before improving, before changing and getting a new facilities, do you think that the CCS can be applied to existing facilities?
Yeah, absolutely. I think it's more important maybe for existing facility, because if the technology is associated to a higher risk, so to a lesser grade of separation between the operator and the product, I think it's even more important to set up a series of controls and mitigations to put the risk down to an acceptable level. So it's not for sure something that will be required only for new facilities. And I would say it's even more critical and important for existing facilities.
Okay. So if there are no other questions coming up, I would stop here. Sorry, let me check. No, I have some questions, sorry, just a couple of them. There is a question of what should be, in your opinion, the real level of details in the real level of details to be included in the CCS document. The level of details to be included in the CCS document.
Mm-hmm. Yeah.
So important.
Yeah. Of course, it's not easy to
generalize, maybe.
To generalize. Of course, CCS document is an overall document, so it cannot go into the very, very specific detail, but I think it should be harmonized with and should be the basis that drive the issuing of the detailed document. So it cannot go into the detail of how you validate the sterilization cycle for your filling part, for example, but of course you should find in the CCS the principles and the guidelines for issuing and for managing this validation. So it's very difficult and dependent by the specific process, but I think it should contain a level of information that can allow you then to develop the specific document in a good way, in a compliant way.
Okay, there are a couple of other questions, but I think that it would be better to let our sponsors to show what they would like to present to our participants. So I would invite Commissaire as a doctor, as a first.
Yes, I'm here. So thank you for this chance. So I would try to give a little bit of the understanding on what we propose and have in terms of our portfolio for the application in the sterile processing environment. Therefore, I will start here, and then eventually if there will be questions, I'll be happy to take those. So first of all, just a brief overview. I've seen also there are some questions over there where we act in terms of isolator technology and all the things which relate to isolator technology and in general barrier systems. In the API processing, the fill finishing, the QA microbiology, and ultimately also in the compounding pharmacy and ATMP, which I'll touch base very quickly at the end, which will be advanced therapy medicinal products. So in these terms, as we know, the upstream on the API, we perfectly know, and Francesco before also had to mention this in the revision in one of the comments, which I fully understand, relating to the high potent handling of substance, which requires protection of the operators, and therefore all those kind of process which relate to highly potent APIs. So we have a certain classification, as you know, maybe 5 or 6. The fact is that in certain instance, also this kind of requirements is also meeting the aseptic processing. So it's the kind of application in some hormones manufacturing is one example. And therefore, the isolator technology itself to this kind of application is basically a unique, defines a unique solution for which the understanding on the containment side, therefore to protect the operator and also to protect the product, needs to be stay in place together. The other one, it's on the sterile API. Therefore, all the intermediates needs to be treated aseptically from the beginning. And this one, it's an application, as we know, it's historical application, but this is where it requires a more holistic understanding also of the whole process in terms of the product itself. And therefore, there could be application in the, of course, antibiotics, but vaccines as well. I mean, this is definitely recently in 2 days, very much a discussion and projects jumping up. And bags, sterile bags, I mean, let's say all the packaging for the active pharmaceutical ingredients. While in the fill finishing, which is probably the parts which relates more directly to the Annex 1 discussion we are having here. So, of course, isolator technology and in general RABs are applied to different application of formats. So there are conventional formats like vial, syringe, cartridges, but as well, and here I think there could be some need of distinguishing isolator and RABs. I feel always there's a little bit of confusion in understanding when to use one or the other. I've seen one of the questions in the Q&A relating to that, and eventually we can come back to that later on. So in this case, there's also, I mean, recently, 3 years ago, we have launched also kind of a campaign called ModRabs for the revamping of legacy facilities. So existing facilities which don't even have RABs actually in light of what the requirements of the new Annex 1 was coming out, to be able to revamp an existing filling line with an open RABs. So apart from that, of course, application on the new projects, and 90% of the time at today, we see customer demanding isolator technology instead of RABs for different reasons. But I would suggest you also, there's a survey which has gone out about 2 months ago, which was done by the ISP, which basically tracks all the trends in the world for which concern isolator and RABs apply to the fill finishing. And therefore, it's a very interesting publication that you can get on the ISP website and gives a little bit of information on this side. Apart from the conventional format, we are seeing a lot the requirements on IV bags and also combination products, which are like, let's say, special devices, ready to mix device, which have the requirements of being treated aseptically, and therefore they jump into isolator technology and all the related automation. One of the things that it's important to mention is that when we design a new process or an existing process, but we want that to be included within the barrier technology to be isolator, we need also to sometimes redesign all the automation if in this case are like customized devices. And therefore, in this sense, what the whole strategy on how to make that process fulfilling the aseptic requirements and or containment requirements needs to take into account not only the automation itself, but the isolator and the whole the assembly together. And that's, I would say these ones are probably the most challenging projects and realization that we are called in to make. So, of course, briefly mentioning also the QA microbiology, the sterility testing and glove testing as well. We talked briefly as well, Francesco mentioned the glove disinfection and so on. One of the issues on the new Annex 1 draft is also the glove testing itself, which compared to the old revision now, it's getting basically more as a mandatory statement for the application, either in RABs or isolators. So in this sense, this one for us, it's actually something that we're seeing a lot of demands at today, because of course it's applicable on all the existing plant or new plants and so on. While on the sterility testing, it's more or less what has always been there. We have launched recently a new development project for an automated version of the sterility testing, which we're working on as we speak and will come out later this year. And last but not least, I wanted to mention our efforts in the ATMP. I mean, we know this one, it's a growing trend is actually taking a lot from the Annex 1 in terms of manufacturing guidelines, even if it's on a separate document from the European Commission. And so barrier technology, isolator technology are actually going into this direction as well, and are actually today supporting the development of the ATMPs, which of course, as we know, there are very few of them which are in the already approved and licensed, while there are many which are still going through the clinical trials. But we're actively supporting this with specific product portfolio, and I will invite you to visit the website for an installation which was done in Germany, which corresponds to the largest cell culture plant today in whole Europe. So pretty interesting to check. I would just a couple of slides to introduce my colleagues from the Lama. One of the things where we study the isolator technology, and I think my colleague from the Lama will show that much better than that, is the integration of the autoclaves within isolator technology. So to keep the grade A continuity, this one is an application where we're seeing actually in the last 3 years, a lot of demands in the aseptic processing, either connected to the field finishing operation, but not only, also in the upstream sterile API processing. So with that, I should be perfectly on time to leave it to Guido, I think. Right, Piero? You tell me.
Yes, yes. Thank you, Massimiliano.
All right.
Thank you for your presentation.
Good evening, everybody. And thanks to Massimiliano for getting my lead. As you might know, the Lama is a manufacturer based in Italy of sterilization equipment and washing equipment since the late 40s. So we have been active in the pharmaceutical field for more than 70 years. Where we are connected to this barrier technology, we are connected there since we are able, as Massimiliano showed you briefly, to integrate the sterilization equipment or washing equipment or any equipment manufactured by the Lama into a isolator. Therefore, whatever is the type of process that shall take place in the isolator, we are able to connect it to one of our equipment in order to make it as a sort of satellite to the isolator itself. The story began 13 years ago with the commissure when we introduced at first air steam mixture autoclave into their isolator. It was a unit for radioactive isotopes filling and sterilization. And the unit was specially designed entirely for commissure. And ever since, we had the chance to recognize that a number of other units of the Lama can be successfully integrated with larger units or with different type of processes, meaning that the Lama, as a maker of autoclaves, sterilizers, washers, etc., can integrate whatever unit to an isolator through a specific conceptual design that we named magneto door, which is a magnetic-driven door sliding that do not allow the waste of space between isolator and the process unit because of the swinging door. In fact, the sliding mechanism remain extremely minimized in space in a way that the isolator do not change drastically its shape for the integration. At the same time, the magnetic driven door allows sanitization by hydrogen peroxide as normally occurs inside the isolators. Another interesting part of the magnetic door is that once integrated in an isolator, the rear part of the doors can be reached by gloves and therefore also manually cleaned in case of need. I'm showing you now a video where we sketch and outline the way normal equipment of the Lama, in the case of the video we show an oven, can be integrated into a generic type of isolator. It's a dual door system in a way that we load from one side, as you see in this very moment, the equipment that you want, the tools, the goods that you want to process, and this will be, at the end of the process, downloaded into the isolator for further processing. Obviously, the video shows an oven, but the concept is that whatever type of our equipment can be integrated in the very same way. Considering that the Lama is able to manufacture, as you might see here starting from the top down to the right, saturated steam or air steam mixture autoclaves, hydrogen peroxide sterilizer. When I say sterilizer, I mean log 6 reduction in high vacuum, meaning that is a real sterilization for everything but liquid loads. Washers, decontamination unit with hydrogen peroxide, or combined unit, which combine one of the technique I just shown to you into one only unit, meaning that we can sterilize, wash, decontaminate in the very same equipment. Then whatever of this unit can be integrated with whatever type of isolator using the technology of the magnetically driven sliding doors. So to summarize, we have a sterilizer that can be whatever type. We master also the hydrogen peroxide technique, either in decontamination or sterilization mode. And we master the technique of washing. These three techniques can be either combined together or in standalone mode, and they can be integrated into the isolator. The presentation refers obviously to a concept that is not a concept only, but several units have been already manufactured and sold all over the world with the same techniques. Basically, the idea, to summarize the idea, we can outline three different types of application. In the configuration number 1, the Lama equipment, either a sterilizer, a dry heat sterilizer, an oven, a DHS, or a hydrogen peroxide sterilizer with deep vacuum, which is a technology that only the Lama master in Europe, or washers combined with sterilizer, or even a simple pass blocks, can be the entry deck to an isolator, as we see here on the left side of my screen. At the bottom, configuration number 2, the Lama unit can be vice versa used for a terminal sterilization process whenever is required, exactly like in the case of Commissar. What we did with Commissar 13 years ago is nothing but a regular terminal sterilization process. Configuration number 3 is instead when the Lama equipment is used as an ancillary equipment to the isolator. For instance, as an example, you have your filling line within the isolator. You want to wash and sterilize the machine parts which are needed for the change format, and you load them inside the Lama equipment, and you re-download them into the isolator at the end of the very process. I hope I have been clear enough with my brief sketch of what are our capabilities. Please feel free to ask any question may arise. Thank you.
Thank you, Mr. Rovera, for your presentation. I hope that we were able to present to our participants good examples of innovative technical solutions presented by successful companies, which shows we've shown a good deep competencies in two parts of in two different features. One is the process, so sterilization, washing, and so on, and the other part is containment, and the combination of different expertise in this kind of equipment can be a solution for sure to meet specific requirements, more and more specific requirements. Now we still have some time for we have a few minutes left. If you like to send some questions. I think that we have a couple of questions coming up. I would ask Francesco to reply. What is the expectation of the supervision of the aseptic fillings? Do they need to be supervised by quality personnel, microbiologists, or for any other areas?
I think the expectation is to have a person which is qualified for this role, so which is knowledgeable about the process, knowledgeable about the behaviors that should be applied. So for sure a microbiologist part of the quality unit, I think would be the best solution for this kind of supervision. But I think that the most important thing is that the process is knowledgeable and can provide an effective coaching on the behavior, because it's an important part of the contamination control strategy.
In fact, you also commented about the supervision of aseptic process innovation. Do you think it is necessary to supervise the whole process or just the critical points?
I think, of course, the critical intervention, especially where you have the personnel entering the grade A or moving closer to the grade A, are the most critical part. By the current wording of the annex, it seems that the expectation is for the whole process. So there is no indication in the wording about a specific part of the process. So this is also what we commented. So let's see the final version of the annex. But I think, of course, the higher is the risk of the task, the most benefit you can have by the supervision.
Sure.
But so far there is no text that allows you to focus only on this.
Yeah.
According to our interpretation.
There is a question about, do you see some in the chatter?
Yes. I think the description of the whole facility in the CCS, we already addressed this. Is terminal sterilization line can be used for aseptic filling? Consider the filling machine installed in grade C under LAF A? No, it's not possible, because the expectation for the aseptic process is to have at least a grade A and surrounding grade B. So this configuration is good for the terminal sterilized product, especially the additional protection by LAF is a good thing. And it seems an emerging expectation, but no, this configuration cannot be used for aseptic process. Unless you have an isolator, but this is, I don't think this is the case by the wording of the question.
Okay. So I think that we we should go to conclude this webinar. I have one general question from my side to you, Francesco, just a general point. Do you think that the impact of these new requirements, if they will be totally confirmed, do you expect an impact on mainly on what? On people training or new procedures to be developed, or to change existing facilities, or to an impact on your on the process time? Maybe you have an impact due to additional activities to be performed. Just an overview of the future with the new Annex 1 applied to at the floor side.
Yeah, it's, of course, the Annex is covering all the aspects of the aseptic manufacturing, so there will be impact in many different areas. And some of these will be impacting for the companies. If, for example, the expectation of performing the integrity test of the glove before and after each batch, from a times perspective, for big lines with almost hundreds of gloves, this will be an impact in terms of time. So the companies will have to, also the technology should help the companies to go to a more quick, reliable test, for example. But this is just an example. So it's there is not an area where the impact will be more than others, because again, the improvement and the new concept introduced are in, I think, almost all the aspects of the aseptic process.
I would be pleased to have questions to be addressed to our sponsors. And I've just received a request about which are the difference between the different types of reps.
Yeah, I can take that. So firstly, it's it will be just two kind of wraps. So it's an open wraps. So wraps in terms, just a step back, wraps definition, it means that the environment all outside, it's a grade B environment, and therefore inside the wraps it's a grade A, while the isolator it's a grade C, and with the isolator inside it's a grade A. So if we stay on the wraps, therefore the outside it's grade B. There are two kind of wraps. One, it's an open wraps, which basically have an overspill outside in the environment, therefore the laminar airflow, there are barriers, and therefore there are two barriers, which is the dynamic barrier of the slide over pressure, which in the gray zone can be achieved, as well as a physical barrier so that there's panels. While the closed wraps, it's a wraps which is able to basically close everything, so it can have a decontamination system, so a VHP technology integrated, because it can seal like an isolator and therefore decontaminate only the internal part. From an outlook of technology, how they appear, they're very similar, closed wraps to an isolators. But of course, the closed wraps having and therefore has recirculation and does not overspill the air from the grade A zone to the grade B. The thing is that, of course, having a grade B background, that allows to easy the material transfer inside and outside of the grade A zone. In this regard, however, there are publications from the PHSS, ISP is coming out with one, so therefore eventually we can provide further literature to have some schematic.
Thank you, Massimiliano.
No problem.
There is a question arrived about terminal sterilization. I would ask maybe Guido Rovera, to consider. It is the terminal sterilization line can be used for aseptic filling for the same line, considering that filling machine installed class C and under a laminar airflow class A. I can't understand very well.
Yeah. This is the question I addressed before. So no, it's not possible to run an aseptic process with a grade C environment unless you have an isolator.
Do you want to comment, Mr. Rovera?
Basically, it's exactly what Francesco said. The way you choose to thermally sterilize or not the product depends on the risk assessment. And therefore, even if you are connected to an isolator or if you are not connected, the way you shall choose the integration with an autoclave for terminal sterilization depends really on the risk assessment you run for that kind of product and for the specific application of it.
Okay, thank you. I think that we can now close our webinar. Let me have 1 minute to add some news, because I had a contact with the other organization just a few days ago. I'd like to let participants know that European Commission received about 2,000 comments this time, so not the 6,000 as in the past. It is important to consider that ISPE is organizing an annual meeting, which will be a virtual one, early November, and ISPE announced that that meeting, Annex 1, will be an important topic of discussion with the presence of authorities. So it will be the opportunity early November to know something more about what the authorities are preparing as their final version. I would also add that EIPG, together with the other associations, has been invited to a meeting which EMA, European Medicinal Agency, organizes annually with the interested parties. And this, the date has not been already arranged, but to be before the end of this year. And that would be another opportunity to understand. Based on the last year, the last year interested parties meeting, when EMA announced the imminent issuing of the Annex 1 version 12, they also anticipated that there will be a timing, a definition of timing for the application of the different requirements, but they did not add any specific clarification of details. So I based on what I know in our contacts as a EIPG, I expect that the revised version will be released early next year with a schedule of the application, the entering into force of the different requirements. That is what I could say as a final comments from my side. I want to thank you, everybody. We had more than 300 registered participants. And so I think it was a really successful webinar. Thank you to the speaker, to the two sponsors, for the interesting presentations. And of
Thank
course,
you.
Thank you very much.
Tecniche Nuove for the technical support. Goodbye.
Bye. Thanks.